Good morning, grade 8. How's it going? I hope all of you are doing well. And today I have a new classmate, okay? So she will be the future teacher inside our class, Perry, and she will share me to explain the steps of meiosis. So let's start our session. Okay. So today we'll complete chapter 4, heredity, section 2, meiosis. And you can open your Red Interactive book, page 61. And at the end of the session, we'll be able to identify the steps of meiosis and how the genetic variation happens between people. And before starting steps, I want to remind you how are 6 cells made. So do you remember how are 6 cells made? You can raise your hand. Yes, Jumana.
They are made during meiosis.
Yes, during meiosis. Okay. So we said that we have process called meiosis. And meiosis is a copying process that produces cells with half the usual number of chromosomes. So the produced cells or the 6 cell will have the half number of chromosomes. And meiosis keeps the total number of chromosomes the same from one generation to the next. As we said, during fertilization, the half number comes from mother and the half number comes from father. So together will make 46 chromosomes. So meiosis keeps the total number of chromosomes the same from one generation to the next. And before starting to talk about steps of meiosis, let's enjoy this video, okay? And then Perry will talk about steps of meiosis in details. So let's start.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay, back again. So let's share presentation for Perry. Okay. Perry, are you ready? Yes, Perry.
Yes.
Yeah. We can start now.
Okay.
Oh, come on.
Can you
Okay.
hear me?
Yeah.
Okay.
Okay. So start, and you can open your cam if you want.
Okay. How do organisms reproduce? When organisms reproduce, the parent's genetic information is passed on to the offspring. So as we know, when a human being, they come to reproduce, their sex cells, a female and a male, they fuse to make an offspring. So the offspring gets half of the chromosomes from the mother and half from the parent, which is why the genes are the genetic information or the genotype. Half of it is from the mother and half of it is from the parent, from the father. And there are two kinds of reproduction. There's asexual reproduction and sexual reproduction.
Okay.
Okay, next.
Next.
Asexual reproduction, it happens only with one parent. It doesn't need two parents, it needs only one parent. And asexual reproduction produces an offspring that it has the exact same genotype as the parents. So if like any organisms that reproduce asexually, the offspring will have the exact genotype as the parent, and it might also have the same phenotype. Which phenotype is how an offspring looks. So it might look exactly like the parent, but that's not as high. Like the percentage of that is not as high as the genotype. In sexual reproduction, two parents are needed. One cell from the father, which is the sperm, and one cell from the mother, which is the egg. They fuse to form the offspring. In sexual reproduction, the offspring never looks like exactly like one parent. It shares traits from both parents. It never looks exactly like either of the parents.
Okay.
What are homologous chromosomes? So we remember that genes, what are genes? They are the instructions that carry the inherited traits. So, as I said before, offspring, they take their genes from their parents. Their genes are passed on from their parents. So half of them is from the mother and half is from the father. Genes are always located on chromosomes. Each human cell has a total of 46 chromosomes or 23 pairs. So when two cells fuse to form an offspring, the 23 chromosomes from the mother and 23 is from the father. When they fuse, they form 46, which is the normal number of chromosomes in any human. Now, how are sex cells made? Sex cells are made during meiosis. So sex cells, these are what we need to reproduce. We can't, reproduction cannot occur with normal cells. We need the sex cells to produce. So what is meiosis? Meiosis is a copying process that produces cells with half the usual number of chromosomes, which is 23 chromosomes instead of 46. Because when the egg and the sperm fuse, the chromosomes are added and they form the normal number, which is 46 chromosomes. And that's why sex cells have the half number of chromosomes. Now, the steps of meiosis. In meiosis 1, first, before it begins in the prophase, the chromosomes are copied and the DNA is copied as well. Then the nuclear membrane dissolves or disappears, making the homologous chromosomes clear, and they are more obvious.
We start meiosis, okay, with interphase. And during this process, chromosomes start to replicate or duplicate, okay, in interphase.
Okay.
So preparation
An interphase.
And then we have two steps for meiosis, meiosis 1 and meiosis 2. So what's the difference between them? And we want to know the name of each stage.
Okay.
Yeah.
So meiosis has two stages, meiosis 1 and meiosis 2. It's divided into two cell divisions. So the first one is called meiosis 1. So before meiosis 1 begins, in the interphase, chromosomes and DNA are replicated or duplicated. Then the division starts. The nuclear membrane dissolves or disappears, and the chromosomes are now clear. Homologous chromosomes or pairs line up at the equator of the cell,
Yeah.
which is called the metaphase plate. And then the chromosomes separate using the spindle fibers. The spindle fibers pull apart each sister chromatids, and they separate from their homologous partners. Then they both, each homologous pair moves to the opposite side of the cell. The nuclear membrane reforms. Now we have two cells that are almost divided, but they still didn't divide in the cytokinesis. And on the end of each cell, there are the chromosomes. Then the nuclear membrane reappears, and the cell divides via cleavage furrow, and the paired chromatids are still joined. Now we have two cells, two haploid cells, and they are and the chromatids are on inside now.
Yeah, we have two now. And each one will make
Yes.
the same steps to give two. So we start with
Yes.
haploid cell, okay, and give two
Mhm.
haploid cells. So finally we'll have 4 daughter cells or 4 haploid cells. 1, 2, 3, 4. So what happens in meiosis 2? Starting with haploid cell.
Two haploid cells.
Yeah. So we'll start with prophase 2, metaphase 2, anaphase 2, and telophase 2, and then
Yes.
cytokinesis.
But the difference is that in meiosis 2, the second division, before it starts, the chromosomes, they do not replicate again.
Yeah. So we start with haploid number, and in this case, in prophase 2, no duplication or replication for chromosomes will be the same.
Yes.
Okay. Then after prophase 2, metaphase 2. So what happens during metaphase? The chromosomes
The
line up.
chromosomes yes, they line up at the equator of the cell, and
Yes.
then the spindle fibers separate them again.
Separate them in
And
a process called anaphase.
yes.
You remember anaphase? Yes, they are fight with each other to attract the chromosomes on the poles or sides, okay, of the cell.
Okay.
And after anaphase, telophase, which in this
Telophase.
Yeah, telophase 2.
The cells start to divide.
No, what happens
and
in
the
telophase?
nuclear membrane reforms.
Yes, reforms. So what's disappeared in prophase will reappear again in telophase. So the nuclear membrane will reappear, and like here, construction in the middle of the two cells. And then after telophase 2, cytokinase. Oh.
The cleavage furrow forms and separates the cells.
So let's revise again. We have prophase, metaphase, anaphase, and telophase. Last one, cytokinesis. So again, guys, prophase, metaphase, anaphase, telophase, and cytokinesis. At meiosis 1, we start with interphase, okay? And then prophase 1, metaphase 1, anaphase 1, and telophase 1, and then cytokinesis. Then we'll start meiosis 2 with prophase 2, metaphase 2, anaphase 2, telophase 2, and then final cytokinesis to give finally 4 daughter cells. You want to add anything, Perry?
I'm just going to repeat fast that like explanation of each one so that they remember it.
Okay.
So in meiosis, it has 2 stages, meiosis 1 and meiosis 2. So the difference is that in meiosis 1, before the division begins, there's interphase. So in interphase, the chromosomes or the DNA replicates before the division. So that's in the interphase. In the prophase, the nuclear membrane disappears and the chromosomes are more clear. And then we have the metaphase. In the metaphase, the chromosomes align at the equator of the cell, and the spindle fibers begin to pull them apart, and that's in anaphase. Anaphase, the chromosomes are pulled apart. Each chromosome is on the opposite side of the cell. And then we have telophase. In the telophase, the nuclear membrane reappears, and the cells start to divide slightly. Then we have cytokinesis. In cytokinesis, the cleavage furrow divides the cells, and now we have two haploid cells. That's in meiosis 1. In meiosis 2, there is no interphase. The chromosomes do not replicate before the division. So the division happens right away. So then we have prophase. Again, the nuclear membrane disappears. And then metaphase, the chromosomes align at the center of the cell. And then they start to divide in anaphase. In anaphase, they are now at the opposite sides of the cell. And then in telophase, the nuclear membrane reforms. And in cytokinesis, the cleavage furrow separates the cells. And now we have 4 haploid cells, each one with half the number of chromosomes as the original one. So the original one, it had 46 chromosomes. Now we have 4 haploid cells, each with 23 chromosomes.
Thank you so much. Big clap for you. So we have questions here. Okay, so Jumana, raise your hand. Jumana.
I'm sorry, this was from before. I'm very sorry.
No problem. Okay, Fatma.
Yes, Miss, I want to ask a question about the chromosomes.
Yeah.
Is the homologous chromosomes 2 chromatids or 2 different chromosomes?
Okay. So, Perry, could you answer?
Yeah.
And I will answer after
So
you.
homologous chromosomes is a pair of chromosomes, which means 2 chromatids together, a pair of chromosomes, which it has the same, or like it carries the same set of genes. So it's 2
Yeah,
sister
so they are not 2 chromosomes.
Okay. So listen, guys, because you're always confusing this, what the meaning of homologous chromosomes, okay? So chromosome, okay, it at the beginning, okay, before meiosis or before mitosis, before any division, it starts with one strand, okay? So this one strand called the chromosome, and after duplication makes a sister chromatid, and there are 2 chromatids now attached with at the center with centromere and called chromosome 2, okay? So chromosome may be 1 strand or 2 strands connected together with centromeres, but in this case, it happens at interphase or before meiosis or before cell division, okay? Okay, so what the meaning of homologous chromosomes? Homologous chromosome, as you watch it in the video, okay, like 2 strands of chromosomes like each other, carry the same number of genes, okay? So these called homologous chromosomes. But after duplication, each chromosome make a sister chromatid. So we started in meiosis 1 with 2 chromatids in 1 chromosome and 2 chromatids in 1 chromosome. So we have 2 chromosomes now called homologous chromosomes, okay? Again.
No, Miss, I don't understand.
Okay, let's wait, Fatma. Okay, so here you can see this picture, okay?
Okay.
This picture. So during metaphase 1, we have here homologous chromosome. Sorry. So we have here chromosome with 2 sister chromatids, okay? And here we have another chromosome with 2 sister chromatids, okay? And these 2 chromosomes, total chromosomes, called homologous chromosomes. Why called homologous chromosome? Because they have the same number of genes and the same sets of genes. Maybe these genes different, recessive or dominant, as we talked before, but the same length, the same sets of genes, okay?
Okay.
Is it clear now?
Yeah, it's better.
So homologous means pair of similar chromosomes, similar sets of genes. So here, before duplication, it will be just 1 strand for this chromosome. And this before duplication will be 1 strand. In this case, we say homologous chromosomes 2, okay? So chromosome may be 1 strand before duplication or 2 strands. And in this case, we say 2 sister chromatids and call the chromosome 2, okay?
So both of them are called chromosome. It's okay if it's 1 strand or both 2 strands?
Yeah. Yeah.
Okay.
Okay. Okay.
Okay, thank you.
Welcome. So, anyone have another question, guys? Any questions? Okay. So I want to sum up, but I think we have just 7 minutes. So thank you so much, Perry, for your presentation. And let's sum up again with pictures here. Okay. So during meiosis, we have 4 phases. Okay, during meiosis 1, and cell division that reduces the chromosomes number by 1/2. So during meiosis 1, it will produce 2 cells with the half number of chromosomes. In 4 phases, we start with prophase 1, metaphase 1, anaphase 1, and telophase 1. And you have to write 1 or 2 because it's different between 1 and 2. Okay? So prophase 1, it's longest and most complex phase. So it takes almost of time. And 90% of meiotic process is spent during prophase 1. So you can imagine 90% of the process taking place in prophase 1. Okay, and during prophase 1, chromosomes condense, and synapses, it's a process in which homologous chromosomes come together. So homologous chromosomes, after duplication, okay, come together and form tetrad. So tetrad means 4 chromatids. I hope to see this picture. Yes. So you can see here chromosome and another chromosomes because they are similar. So it's called homologous chromosomes. And because there are 4 chromatids, so it's called tetras. Tetra means 4. 4 chromatids, 1, 2, 3, 4. Okay. And these homologous chromosomes on both sides. Okay. And these tetras makes a process called crossing over. So crossing over, which make variety or genetic variation between people. Why we not look similar to our parents? Because of this crossing over, okay? So crossing over is one of the 2 major occurrence of meiosis. The other is non-disjunction. Okay, so just we have to know crossing over occurs between tetrads, okay, to make variation. So I want to see the crossing over here. Okay, so we have 4 chromatids here, and the crossing over happens between the 2 different chromatids, okay? Different chromatids, not at the same chromosome, no, between the 2 chromosomes. Internally, make crossing over exchange. So we can see here another chromosome with another traits, another variation, okay? Okay, so this one or this cause or crossing over causes variation between people or between offspring. It causes diversity or variation. What may I want to ask? Yes, Fatma. Fatma.
No, sorry, this was from before. I'm sorry.
Perry. Yes, Perry.
Yes.
You want to ask?
No, I'm good. Thank you.
Welcome. Okay, so let's complete. Okay, so this variation due to crossing over. Okay, and then we have question here. A cell containing 20 chromosomes diploid at the beginning of meiosis. So after meiosis, the produced cells containing how many chromosomes? So we start with cell with 20 chromosomes diploid. So after meiosis, it will give 4 cells with how many chromosomes? Who knows? Okay, Fatma.
Maybe I don't know, but like each cell will get 20 chromosome.
After meiosis, we start with diploid cell, okay, 2N, 20
Okay.
chromosomes. So after the second meiosis, we have 4 cells. Each cell have the half number. So half 20 will be 10 chromosomes.
Yeah, 10, yeah.
Okay, you got it?
Yes.
Okay, so remember guys, we start with 2N, cell with 2N or diploid, 20 chromosomes. So at the end of meiosis, the aim of meiosis to give cells with the half number of chromosomes. So the half of 20 is 10. Okay, the answer is 10 chromosomes, which is called the haploid. So haploid means the half number. Now we have another question. It will be the last one. Okay, so a cell containing 40 chromatids at the beginning of meiosis would at its completion produce cells containing how many chromosomes? So we start with 40 chromatids. So how many chromosomes? The produced cells with how many chromosomes? Who knows? So you can think about it. What's the meaning of 40 chromatids? Okay, let's take Perry. Yeah.
40 chromatids means like 20 chromosomes.
Okay, 20 chromosomes.
Like
So,
20 pairs of homologous chromosomes.
so the answer will be
10.
10. Yes.
It's half of 20.
Yes, because 40 means the 20 chromosomes at the end of meiosis will give us or will give cells with the half number, haploid number. So it will be 10 too. Thank you so much. Okay, so we have now just 1 minute and I wanted to say thank you so much. Okay, and please, I will revise the lesson, okay, next session, inshallah. And thank you so much, Perry. And let's see the video together. Okay, so thanks for watching and enjoy this video. Bye.
Nope. Yep. 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 Nope.